To even think this is going to cause weeping and gnashing of teeth for some people in various parts of the scientific community. How could I even ask that, they would think? How dare you? Well, you've got to hear me out. You've got to listen to what I am suggesting, and then you can refute it afterwards. Is that reasonable? I will explain to you why I asked that question and why I think it's important for us to reflect on in case we have another problem coming up again in the near future. So that's all I ask. And I'll explain to you why I've come to that conclusion. You don't have to agree with it, but I'll explain to you the science around why I've reached that conclusion. And I'm confident with regards to talking about the science because I'll show you this here is that I think I'm the only person in the world who has put together a comprehensive COVID overview. And this is from my scientific perspective. This is just an example of all the slides that I have created in order to produce this course. We're talking about over 200 slides that I've had to put together. So it's a very unusual position in terms of making this happen. And this is the 360 course that I did. Now, you can argue about my opinions on it, but you can't argue with the fact that I have actually really gone through the research to come to these conclusions. That's what I'm saying. So hear me out on it and then make a judgment as to whether or not what I'm saying makes any sense whatsoever. The first thing that I will clarify is that I am talking about injectable vaccines for COVID 19. Okay? I'd always said early in the pandemic that what we needed were mucosal vaccines, but all of the money and everything was going to injectable mRNA technology and so on. I think that was a mistake then, and I think all the evidence now indicates that it probably was correct. So I'll explain why, because this is not about other diseases. This is just really specific to COVID 19. And part of the problem is that much of the scientific community has jumped down the pathway of trying to find an answer to a disease they don't understand. As I think it was Abraham Lincoln was saying, if you're going to get him to cut down a tree, he will spend, if you give him 6 hours, he's going to spend 4 hours sharpening the axe. That's how we should have been. Study the science, make sure it makes sense before you then go and approach trying to solve what could be a very complex disease. Just a reminder before I get into this, remember coming up in a very short time is session 3, Health and Wellness Network. And I can be a little bit more candid in these sessions, talking about different aspects of the pandemic, different parts of health. A lot of people are struggling and they don't really know what to do. And I'm trying to see if I can add some insight from a clinical perspective as well as a research perspective to help people to understand it. You can register for free. There are donations as well. So please click on the link below and join us. We have quite a crowd with us, and I'll be able to try and bring some more concepts to you. But let's get back to the question. Why did I say what I said? And what is my perspective on the science? And again, many people may disagree with it, but this is just what I am thinking about. So let's take you through a few slides to show you my analysis of the science. So, just from basics, always starting with the virus. As usual, this here is your virus, and on the surface, the blue bubbles that are three together. This is the spike protein. You have these little red dots, which are membrane proteins, and you have a little orangish dot here, which is an envelope protein. And if you cut through the virus here, and this here is the capsule around it, and inside you have the RNA, which is then what makes all these proteins, and you have a nucleocapsid protein in here as well. All of these proteins that are highlighted are important because this is how the immune system tends to target the virus. Now, before I go any further, I'll tell you something that's very important is that actually for natural immunity, it doesn't really focus so much on this spike protein. It focuses on some protein sequences that are inside this mRNA called ORF sequences. And that's the primary thing that it tends to look for in terms of developing immunity, which suggests that it's not really antibody, it's more T cell immunity that the body uses to fight against this virus. But yes, that's the basic point with regards to where we are with the virus. The next thing that you have to understand is that about mucosal immunity. Now, as I said, I'm jumping through some of these points. There's a link for the full course. This is almost 60 modules, short courses with sections so that it hopefully makes it easier for people to go through. But the mucosal defense is exceptionally important with this virus. It's important with every virus, but this virus is special. And what it's showing you here is that this represents your airway where the air is. And this lining here prevents the virus from getting through. And even if it got through the cells, there is also an immune defense layer that it has to pass through. And this mucosal immune defense is exceptionally valuable at fighting not just COVID, but every virus, influenza, other coronaviruses, rhinoviruses, streptococcal infections. This is a very sophisticated layer of immunity. And the way that I tend to describe this mucosal defense is like skin. If I peeled away your skin, there would still be an immune defense there. But the layer of skin, the protection, the physical barrier of the skin is very important in terms of immunity. So it's the same with the mucosal defense that I describe like the skin inside the airway because the virus can't get through. Now, this is very important when I say the virus can't get through because you have to understand that all of the problems that exist with COVID are when the virus gets into the bloodstream, right? Really, really important point. So, this mucosal defense that we've got here, this is what this black line means, and this represents IgA antibodies lining this here with mucus, preventing the virus from getting through. But if the virus breaks through this, it triggers the systemic immune response. So you can almost think of this as a fence, a big border fence, and they have border guards. But if you break through the fence and get past the border guards, then you have a whole army that will take you on. The problem is that this army can tend to overreact to this virus, and this is what causes the cytokine storm. Really important point. So, preventing the virus from getting through this is really important. But here's what we have done, and this is where I said that we have to think carefully, is that by using an injectable vaccine, you're training systemic immunity and not mucosal immunity. Really important point. Think about that again. So you have your border fence, you've got your border guards, you're training instead of the border guards and instead of training the building the fence, you then just get lots of police and the army just patrolling the streets. So what will then happen is if the terrorist gets into the city, by the time you start shooting at him, you will put other people at risk. And that's the difference between systemic and mucosal immunity. It is always better to keep the virus out. That's part of what I'll be covering as well in this wellness course. Is that I'll get a bit distracted with this just for a moment, but just to tell you that there are two sides of the immunity problem. The first side is that what you have to do is try and prevent virus from getting in, but if the virus gets through, you then need to actually stabilize the immune response. So one side you need strong immunity, the other side you need stable immunity because this is what drives all of the responses that cause problems around COVID. But let's get on. When you look at the antibody response to infection, and this is a very important slide, I want you to think carefully about this. When they looked at the antibody levels to infection, they found that on average, the level is at about 280 to 300 nanograms per milliliter. Okay, so this is after somebody has had an infection. So the antibody level, this is what you would call the average or normal response to infection. Some are a little bit higher, some are a little bit lower, but generally this is about where it is. Now, my perception is that I believe that the immune system is perfect. If you are normal, you have an almost perfect immune system. Doesn't do too much, doesn't do too little. It just what's right. And so I always think that the natural immune response is the best template to try and work out what else we should be targeting. Okay? What we found happened with regards to the vaccine immunity is that it had a very, very big antibody level. Remember, the levels there normally was about 280 to 300. Look what happens in the context of vaccine immunity. This is from 2022, but it's still relevant. The principle is still there. They're looking from age 16 to 24 all the way to age 80. Now, what you have to understand is, what do these colors mean? Now, 179 to less than 800 nanograms per mil is this light blue here. So the very bottom here is less than 179. So you could almost say that anything that's below this is actually a little bit less than what you would expect for a normal immune response. But it's not much higher than this. This is where it would be. Just go back and let me just show you again so that you can see this. Look, this level here is at about 280. The one I was showing you was 179. So that's the bottom level that you would look at in terms of antibody responses. So let's go forward again. So down here is where you would consider below this, they don't have a strong enough antibody response. Above 800 is already twice what you would get with a normal infection. This line here is actually 2000. This line here is 4,000. This line here is, well, this is the 4,000 line, and above it is 6,000 nanograms per mil. Remember, this is a 16-year-old who technically should have an immune response down here. Potentially, a proportion of them, almost 10% of them, are having antibody levels above 6,000 and still getting infected. When you look at the age 80, you can see here that almost 40% of them will have an antibody level above 6,000. Now, I take you back again so you haven't gotten lost. This is where it normally is, between about 300. Some people go up to about 800. You can see here 800 there. Down there, less than 100. So the vast majority of people are in that vicinity, which is why they had that 179 to 800. So you having exceptionally high antibody levels, and people are still getting infection. Why would that be? Well, the simple answer is that you haven't trained the mucosal immunity. It's like you've decided to burn the skin and give antibiotics by injection. You can't effectively fight an infection that way. It doesn't make sense. And so, with that in mind, you then realize that stimulating the systemic immune response is not necessarily the best thing for this virus. Okay, now I'm going to take you one step further with the science. And this next step is really important. So, work with me on this. So, one of the most important pieces of research came out in 2016. I always make reference to this. I got a chance to speak to the professor, and this research was critical to understanding what happens with SARS-CoV-2. What you have to realize is that this virus is remarkably good at evading your immune responses. Okay? And what this image is showing is that green represents the viral replication, blue represents the interferon response, and each one of these is down to three different categories of patients. Now, the reason that mucosal immunity is so important, before I come back to that, is that the virus is probably one of the best viruses we've ever seen at evading the interferon response. The interferon response is the signal, the alarm system that goes off in your body that tells the body that there is virus around. This virus is like a ninja or a panther slinking around in the nighttime, but it is its most effective strategy. It doesn't really do a lot more that would cause problems at the mucosa level if the lights are switched on. So if interferon switches on, it is not even as bad as a cold. It doesn't even cause a runny nose. This is why so many people were asymptomatic, because if interferon is switched on and the virus comes in, straight away the body switches it off and can deal with it. So let's come back to this image here, really important piece of information. If your interferon response is switched on, even though you still have viral replication, it comes down, you don't get too much inflammation in the lungs. Very important to know, you still get inflammation in the lungs, but it's not too bad. Early interferon response. What was interesting is that they found that this was in mice, but the principle applies in terms of the mechanism. If you had no interferon response and a big surge of virus, you still had no severe disease. So the virus is not what is causing the severe disease. What is causing the severe disease is a delayed interferon response that triggers the cytokine storm that leads to very severe lung disease and potentially death. This is the bit that is really important. Because if you can interfere with this process, you can potentially stop the severe disease. So, this is where it gets to be a problem for people who have a very specific narrative. Because what I'm saying is that actually it was demonstrated very well by Dr. Shetty, over 14,000 patients. He used an early treatment strategy. None of his patients died. And it was just because it was interfering with the mechanism that causes the cytokine storm. What he found was histamine and high dose steroids shut it down. Now, because that was not accepted and because that was ignored by the scientific community, they gave you no option. Therefore, your only option was just to have the vaccine. There was nothing else they were giving you. You had to get sick before you would even get anything else, an antiviral in hospital. That was the problem. But even in the context of an injectable vaccine, it couldn't stop the infection because it didn't train mucosal immunity. Now, I'm just going to give you a quick review of all of the candidates here. These are generally, you have this is the mRNA platform. The spike protein is put inside a lipid nanoparticle that's injected, and then it causes the body to get an immune response. You can then create a DNA plasmid with S protein, or you could just inject the S protein. That's another strategy, the spike protein, to get an immune response. The adenovirus vector platform, this is the AstraZeneca and the Johnson and Johnson, they put the DNA inside this adenovirus, which would then go inside the DNA of the inside the nucleus of the cell, then make RNA and then make spike protein. And then you had the other version, which is the whole virus inactivated, where they got the virus, they then grew it in a cell culture, then they inactivated it, and then they injected this inactivated virus to trigger immunity. And those are the main methods that they used in order to control the pandemic. The problem is that every one of them was injectable. And so, therefore, it was training the systemic immune system and not the mucosal immune system. Now, some people will say, well, I'm sure it had an impact. Well, guess what? We have a patient in Germany. Now, don't ask me why he did this, but this guy was so focused on the vaccine that he got himself 217 doses of the vaccine. Now, there are many people out there who are very much in agreement with this, and they will probably continue to get doses until they've gotten over 200 or 300 doses in a few years' time. But the question is, does it actually have an impact where it needs to impact, which is on mucosal immunity? Well, look at the results from this paper here. Really interesting. So this is showing in black, this was the control group. In red here is the person who had 217 doses. So he had 5 times higher IgG1. He had about the same level of IgG2 antibodies. None of them who were vaccinated, this was a vaccinated cohort, had any IgG3, which is a problem because this is an important part of the immune protection. And both of them again had IgG4. This is the immune tolerant antibody. But when you look now at this here, this is again the same vaccinated cohorts, the normal boosted and the guy who had 215 or 217 doses. His IgG level here in saliva, okay, was much higher, 1.3. There was no response to IgG in the saliva from the vaccinated and boosted cohort. There was no IgM for either of them, but critically, there was no IgA. So the fact that IgG is in the saliva, it's because his levels are so high, it's spilling over. But what will happen is that as the levels go down, the IgG levels will fall, and he'll still not have an IgA response. And this is what brings us back to the issue of the fact that injectable vaccines are not training the mucosal immune system to be able to fight against the infection. That's the drawback. And this is why I said so. What I was saying was that I don't think injectable vaccines are going to work with this virus. This is what I said in 2020, in 2021, in 2022. I got censored for saying that. I mean, it's just the science. Again, just reminding you of the mucosal immune defense. It's very sophisticated. It's combined with mucus here, IgA to block the virus. Then it has T cells on the hair. So even if a cell gets infected, it takes it out. All kinds of systems are in place to protect the body from any virus getting through. Very sophisticated system, but it has not been trained. And actually, the truth is that it has been so immune focused on the spike protein that I think that it is technically damaged, which is why we're seeing so much virus circulating in highly vaccinated regions. So, the reality is that it did have an impact on severity of disease. A really important point. So, just in case people are jumping up and down on the anti-vax side, you do have to recognize that it had an impact. But the impact it had could have been through immune suppression and IgG4. The question then remains is would we have been better by following a strategy like Dr. Shetty, using immune suppression with antihistamines and other repurposed drugs, which I can't mention, to try and block the cytokine storm? What would then have happened is that everybody would have been exposed. Everybody would have then had the opportunity to get natural immunity. It would have trained your mucosal immune defenses, and the virus would have stopped circulating. I keep on asking my audience who are all over the world, in low vaccinated regions, pandemic is over because they all have natural immunity and the virus doesn't circulate so well once that has occurred. But in highly vaccinated regions where you have focus on spike protein, there is a difficulty for the immune system to cope with the variants of Omicron. Inconvenient information, but true. So, just a reminder before I end, remember the health and wellness webinar. Join me there. We'll be able to go through further ideas and questions. And if you want that 360 course, if you really are interested in the science, almost 60 modules, 59 to be exact. I should add another one to make it 60, but it covers all of these things with the images and the breakdown. Those slides that I used were from this presentation. So it's just giving you a taste of what is there. So, thank you all very much for joining me, and I look forward to sharing more valuable information in the near future.